In the last lesson, we have learned the digestive pathway of protein and carbohydrates. So, in today's lesson, we are going to learn the digestion of lipids. This is Stitch. He loves to eat butter, and butter is made up of lipids. So now, let's investigate how can the lipid be digested throughout the alimentary canal. After lipid enter the mouth cavity, it will go into the esophagus as bolus. Peristalsis takes place to push the bolus into the stomach and become chyme. Do you think there's chemical digestion of lipid happening in the stomach? Let me give you some hints. What enzyme is present in the gastric juice? Right, that's only protease, which can only break down proteins. Therefore, there is no lipid digestion in the stomach. Now, this lipid enters the duodenum. This is the liver, gallbladder, and pancreas. They all participate in the digestion of lipid. The pancreas secretes pancreatic juice, which contains all three kinds of enzyme, including amylase, protease, and also lipase. Lipase break down lipids into glycerol and fatty acid. Imagine these tiny blue dots are lipase. They are secreted into the duodenum and start digesting the outer part of the lipid. However, you can see that it is not so effective as the lipase is too small when compared to the big lipid drop. Therefore, let me introduce another friend which can help in facilitating the reaction of lipase. Bile. Bile is produced by the liver and stored in the gallbladder, then secreted into the duodenum through bile duct. Bile contains bile salts, bile pigment, and sodium hydrogen carbonate. Bile salts can emulsify lipid into smaller droplets and facilitate digestion. Here's a jar with water and a layer of oil. When we shake the jar, can you still observe the two layers? Can you suggest what happened to the oil? Right, the layer disappears because the oil becomes smaller droplets and mixed with the water. But can you predict what happens when we leave the jar for 5 minutes? Will the solution remain cloudy or will the oil form a layer again? Here comes the result. The water and the oil form separate layers again. This is a model showing that oil droplets will join together naturally. However, when bile salt is added to the lipid, oil will not join together. How does it work? To give you a hint, bile salt has a hydrophilic head and a hydrophobic tail. How can it separate the oil droplets? Which of the diagram is correct? This is the model showing the correct answer, that how the bile salts stop the oil from joining together. Therefore, bile salts can facilitate the digestion by increasing the surface area for the lipase to act on. After understanding the function of bile salts, can you now answer me whether it is a chemical or a physical digestion? Does the lipid change into a new chemical substance? The answer is no. So, bile salt is a physical digestion. The oil is only changed to smaller droplets without any chemical change. To conclude, digestion of lipid only takes place in duodenum by the chemical digestion of lipase and the physical digestion of bile. I have a final question to ask all of you. Do you still remember what is also present in the bile except bile salts? Do you know what is the function of sodium hydrogen carbonate, and is there any function for bile pigment? To give you some hints, you can think of the pH value of sodium hydrogen carbonate and you can search the color of bile pigment. You can express your prediction by drawing related diagrams with explanation and bring it back in the next lesson.